aeruginosa. And how thick mucus is developed? This is due to the abnormal transport of ion and water. I hope you can see the correct flow of uh, steps in cystic fibrosis disease. It should start with mutation in CFTR, followed by abnormal transport of ions and water, then development of thick mucus, and finally microbial colonization by Pseudomonas aeruginosa. Okay, any questions so far regarding cystic fibrosis?
No, doctor.
No, doctor.
Okay, moving on to the second disease. Generally, it is classified as protein misfolding disease. In this PowerPoint slides, we have three examples for our discussion, namely TSE disease, Alzheimer disease, and Parkinson disease. Others are not discussed, but basically they are characterized by the problems in protein structure. We focus our discussion on three diseases only: TSE, Alzheimer, and Parkinson. As I mentioned just now, the newly synthesized proteins need to be folded into 3D structure so that they can function as membrane protein, they can function as enzyme, they can function as other structural proteins, and many more. If they are not folded or incorrectly folded, they could not function. Then when they are not functional, they should be removed from the cell. They should be degraded, usually performed by protease enzyme. So the problems related to protein misfolding happens after synthesis of protein, after translation. In this case, abnormal protein can lead to formation of aggregates and amyloids, subsequently plaques, which can form holes in several organ, especially brain organ, and finally can lead to death. Okay, the first one is transmissible spongiform encephalitis, TSE. Quite long and quite difficult to pronounce it. It is fine if you just put it as a TSE, standing for transmissible spongiform encephalitis. So this disease happens in brain organ. TSE happens in brain organ, characterized by a myeloid formation, plaques formation, and holes, hole formation in the brain organ. So this abnormal brain structure can lead to loss of control, dementia, and if it is not treated very well, can cause disease. Can cause death. TSE disease in brain organ is related to what we call scrapie protein. Okay, so scrapie protein is actually a kind of protease-resistant protein. PRP is actually normal protein. Okay, and due to the problem in the structure, uh, it produces a slightly different form of protein, which is called scrapie prion protein. Okay, PRP, protease resistant protein, is simplified as prion protein, and abnormal prion protein can be produced in TSE disease. Okay, using Western blotting analysis, uh, researchers compare between scrapie sample, I mean the sample taken from patient with TSE, and sample protein from normal individual. So clearly, we can see the expression of abnormal prion protein or scrapie prion protein in the patient sample, and its size is approximately 27 to 30 kilodalton. Okay? To further confirm that this protein band represents abnormal prion protein or scrapie prion protein, amino acid sequencing is performed, and comparing the sequence of amino acids between patient and healthy individuals, we can see that that particular protein band is actually scrapie prion protein. Okay, PRP, as I mentioned just now, prion protein can be normal and also abnormal. So the normal PRP is assigned as PRPC. PRPC refers to normal and non-pathogenic prion protein. It is produced, it is expressed in brain organ. When abnormality happens on this PRPC, it becomes PRPSC. S letter is added to indicate scrapie. So PRPSC refers to abnormal prion protein infectious, then can cause TSE disease. So infectious here is not related to any microorganisms or yeah microscopic creatures, but it refers to the condition where, for example, one PRPSC molecule can change, can convert other normal prion protein to become PRPSC. Okay, both PRPC and PRPSC have same amino acid sequence. The difference between PRPC and PRPSC is observed at the structural level. Uh, their conformation or shape is different. So different conformation or different shape can lead to aggregate amyloid formation and subsequently cause plaques in the brain organ.